Okay, hello and welcome to Module 4 of our single-cell RNA-seq analysis bioinformatics bootcamp. Today, we're going to be talking about what to do after you've already completed your clustering, so how to interpret your clusters, how to assign cell type labels, how to understand the biological processes that are going on inside of them. And we're also going to talk a little bit about pipelines that you can use to analyze single-cell RNA sequencing. So before we jump into all of the actual biological interpretation part, I'm joined by Wes, who has volunteered to show off an alternative to the scRNA pipeline that he actually uses. So with that, Wes, you wanna go ahead?
Will do. Gonna share my screen. Boom. So I just want to thank Henry for inviting me to share a little bit some tips and tricks I've learned along the way. I'm not going to go through my entire pipeline because that would take too long and take up too much of this class, but I will go through some of the ways you can do this stuff outside of scRNA and some tricks that you can probably work with, even if you use scRNA, if that's going to be your bread and butter. Some of these tricks will even work with it, and I'll show you how that works. Luckily, Henry did an amazing job in the last 2 weeks explaining how all of the math works, so I don't have to. I can just run those quick lines of code, and you guys already know exactly what's happening in the background. I will explain things kind of as we go, and might move a little quick, but I'll stop to talk about the stuff as some of the stuff takes time to kind of process.
Before you jump in, I just want to say something really quickly. So I noticed you're on the Ubuntu version. Yeah, I was about to ask that. So you're on the Ubuntu version of Zoom. So just a couple caveats with that. When you're in screen share mode, it can be lower resolution for some reason. So would you actually mind zooming in just a little bit so we can, because right now it looks very pixelated.
Yes. So part of that I think is because I'm in the browser version of Zoom. I'm not in the. I don't know if there is a Linux desktop version. But I can make my text bigger maybe.
Yeah, there is, but it's probably, to be frank, it's probably not any better.
Fair. Sorry, yes, guys, I am a Linux user. So hopefully that's not too bad. Does it look any better now that I've zoomed in?
That's much better. And just to remind everyone, you should be following along in your own copy of this, which you all have in the Module 4 folder. So just as a reminder. But yeah, that looks a lot better.
Yeah. You're not going to be able to run everything, guys, because I'm not using a public data set. I'm using my own data set because I want to show you what dirty, raw, real data looks like and why some of the tutorials you follow online are great when your data is pretty and pre-processed, but not so much in real life when you're in the lab and maybe your cells are sitting on ice a little bit longer and you get more cell death than maybe the experiment online does or so forth. Just as a reference, I'm Dr. Wes Wilson. I'm a tumor immunologist and bioinformatician at the University of Pennsylvania in the CCI Cancer Center for Immunotherapies group. We do CAR T-cell therapy for cancer patients at various clinical trials. I've been doing bioinformatics since about 2010, but back then it was all like microarray stuff, but I've only done single cell since 2018. All right, let's do this. So I'm just going to load some of our libraries here. The second one here being single cell experiment. The single cell experiment is basically the life force that is the bioconductor universe for single cell experiments. This package handles all the kind of things that you probably think scRNA kind of handles, even though scRNA's more a visualization tool that's just sort of included open source packages and CRAN packages and bioconductor packages from version 1 all the way to version 4. You can use cowplot and use this for graphing. It's not important. If you like using patchwork instead for doing your graphs, use patchwork. They're all just ways to level out ggplots and put more of them on the other ones. Tidyverse to do my little scripting, data wrangling. Nothing super fancy there. I was a Dipler user for a very long time, but I've only really committed myself to actually going full tidyverse recently. These are some custom scripts. They're not important. I'm not even going to load them. I was copying and pasting to make this kind of tutorial from my actual working scripts, and I use them in other things. So this is a parallel multi-core parameter they'll be using in multiple aspects going forward. And this just tells bioconductor packages how many cores to use. So as Henry's pointed out, some of these things can take a lot of processing power. And some of these scripts are originally designed to be unfortunately, is a very, very old language written on a very, very even older language S, I believe it's based on S from the 80s, definitely designed to work on single-threaded, single-core processors. Multi-core processors didn't exist. So they can take a really long time. So this kind of gets around that and bypasses that for the bioconductor packages. Now, don't run, if you don't have 24 cores, don't run this. Change 24 to something else. If we take a look here, screen fetch, you'll be able to see that I have a Threadripper, so I have 32 cores going on here. So I have plenty to play with. I'm going to run 24 in this. Gives me a couple extra cores to run Zoom and other, browser tabs and shit. Oh, sorry, should I not swear in this recording? Sorry about that. All right,
No
so first thing we're gonna do is we're gonna read in our data. This is a real data set. And one thing you'll notice is that I'm gonna be reading in the raw data, not the featured one. And I'm gonna show you why, because this is kind of an important thing. This happens with my B cells when they die. This comes in with all kinds of stuff. And I'm gonna read in what, Cell Ranger would have outputted as well. I'm going to do a comparison. So what is a cell, right? So before we even analyze cell, before we clean up our data, what are we going to count as these gems as being actual cells? So I've read in my data into this raw kind of thing. And if you look at it, it's now a single cell experiment class, and it has 36,000 genes and 700,000 gems, or what could be cells, right? They're obviously not all cells. We expect around 10,000, right? So I've also read in here what this is from the feature barcode output from Cell Ranger. What Cell Ranger says is a cell, right? So if we look kind of at that, Cell Ranger is saying that, of the 700,000, 9,000 are true. All right, so that's what Cell Ranger thinks. That's great. Cell Ranger 5, which is the version I ran this data on, uses a set of genes, kind of like a mini database, and it basically correlates that to if all these kind of cells match these profiles, then they're probably cells. But the old way Cell Ranger used to do it is it used to basically take a cutoff. Henry showed us those rank bar plots from before. He showed us that bad one, and he showed us that really sexy one. And generally speaking, your real data is going to look like something in between. The old version used a threshold cutoff, and single cell experiment, the package we're using, actually has that algorithm built in. It's called default drops, and it'll use that same cutoff. If you wanted to recreate that method, you can do that directly in there, and we'll do that right now to do a comparison. So I'm just going to create a cutoff there. Now you might notice that I'm adding this back into my raw, so I have everything's in one spot. I made this as a list because I normally handle multiple samples at a time. But this obviously doesn't have to be a list because we're just doing one sample. But if it was, you just put your next one there and so forth. You just put a comma and add more. So some of the stuff's copied and pasted from my actual working stuff. So I'll change things as I go, and I'll upload the final version if I've made anything else. So the next thing we want to do now is we talked about how many were here before, right? So we know that we had 700,000, 36,000. One of the things that's not removed here is the zeros, right? We talked about there's so many zeros. So we want to remove any genes that have zeros all the way across, and we want to remove any samples, any gems that have nothing, any transcripts detected at all. Those are going to be empty gems, right? So we're going to remove those with this one line here. Now we're going to do the empty droplets. So the way I do empty droplets, and the way that's kind of the gold standard of empty droplets, and this has kind of been designed in 2019. It was published, but it had been done before that by Aaron Lunn had kind of published these papers first, is if you assume that the vast majority of that 700,000 of gems are empty, right? If we expect 10,000 cells, that's only going to be 1.1% of our cells are actually of the gems are actually cells. So what you can do is you can kind of do an approach for trying to find outliers. And anything that's an outlier from the mean, from the sort of the pool of RNA that was floating around and picked up in those gems that were from dead cells, from fractured nuclei, the mitochondrial genes from dead cells, anything like that, if anything deviates from that pool, that's probably actually a cell, right? So if the majority aren't cells, then the outliers are. And so that's kind of how this kind of algorithm works. So we kind of create our threshold. I won't go through all the math because for time's sake, I know Henry has a lot of stuff to talk about and I don't want to interrupt that. But I'm just going to be creating here some, and you can go through this code on your own later, creating some QCs on these ranks. And then we're going to create, from that, we're going to create our rank plot, which is what we did last week. So I won't need to explain this because Henry did it last week. I stole this, the color palette from Luke Zappa's 2018 publication. So I want to give credit to him where that's due, but I have actually updated this to work with bioconductor packages on my own and to work with modern packages because things of scatter and scRNA don't work the same way nowadays. But otherwise, you know, I'll give credit where he designed the colors. And so as you can see here, you can see our ranked bar plot now. And it's not as nice as some of the nice ones are. The top looks pretty good. You get this little, after the inflection point though, it's kind of a little gnarly, but you can kind of get an idea of where we kind of think these good cells are and where these bad cells are. Ooh, my radiator thing's making a lot of noise. So we're going to go through this now and kind of remove those empties. So always good to set a seed for reproducibility. Some people like to use the date. That's a good way to do it. It's actually a better habit, actually. So that way it's actually introducing some sort of randomness into your seed generation. You're not throwing your bias because your date will change every time you're running something. As long as you're saving your code and doing version control, then you can always go back to whatever that date was. I tend to throw in leet a lot. And we're going to set our FDR at 0.01. Now, in real life, I actually do 0.001. I'll do like 0.01% instead of 1%, but I want to show something. So I've purposely done this at a different false discovery rate, the FDR here. And this is all done through the empty drops package. So this is all going to do all the math all by itself. The only thing you have to enter in is kind of what you want your FDR to be at, and obviously how many cores you want this to use. And this will take a second to run. Then what we're going to do then is kind of take a comparison look. So this is going to look at all those outliers, what it thinks are cells based on. Ooh, I got an error. Errors aren't good. I didn't run that line of code. So while this is doing its thing, it should anyway do its thing. And it should then be doing all those outliers. Then we can compare what a cutoff finds, what outlier detection decides is a cell, and what the Cell Ranger 5 database approach fingerprint of what a cell is. All right, that's done really quick. A lot of cores. Can't go wrong with that. So this is just passing back that back. I'm just basically saying, hey, if it's not a cell, I want that to be false because the others are already true. Now I'm just storing these values that we just calculated back to my original single cell experiment. So again, like SRAT objects, we can add metadata to your SRAT object. You can add in as many variables, as much metadata as you want back to your single cell experiment object. That's all I'm doing there is just putting it all back in. And if you take a quick gander here, it looks like that we want their true trues. This approach has found about 8,000 cells. So we knew that we had about 9,000 from the first Cell Ranger. Now we have 8,000 from empty droplets. And again, I've stolen this kind of data representation approach from Luke as well, because I think it's really cool. I don't do this for my workflow, because I only kind of process it. This is visualization, just a teaching thing. So just, you guys don't need to do it, but hopefully you'll get the point across. And that is this figure here. So you can kind of see here now on the right that the old Cell Ranger cutoff method, the drop method of doing the outliers, and Cell Ranger 5 found 5,857 cells that were all the same. So they all agree, all methods agree that these are cells, right? Cell Ranger 5 found another 2,000 cells that nobody else found. It said, hey, I think these gems are actually cells. Then Drop and Cell Ranger found another 1,800 that the first Cell Ranger method failed to find. It said, we think these are cells. The Drop method found 1,600 cells, or gems, sorry. It's all theoretical, right? Gems that it thinks are cells. And then both Cell Rangers found 5 that the Drop method missed. So with this going forward, what do you pick from, right? This just gives you an idea that all these things could be cells. And we're going to do cleanup. And Henry's already talked about how you remove dead cells with dead mitochondrial. There's ribosonal ways to figure out if things, depending on how you know your cells died, if they're apoptotic cell death or necrotic cell death. you can roll them out that way. There's going to be cleanup further down. So what we're going to do is we're going to take all of them. We don't want to miss important cells. We don't, especially if they have really rare subtypes, right? Maybe Cell Ranger's algorithm for gene expression doesn't affect your experimental type. Maybe your treatment is different. Maybe you're using weird cells that no one's ever discovered before, and you don't want to lose those because they didn't match some predetermined algorithm. So what I do is I take all of them. So I take all of them. So it's going to be more cells than are probably going to be there, but they're going to get ruled out once we remove dead ones and other stuff like that. So all I'm doing here now is I'm creating a new group called selected. It's a combination of both of those types. It's going to be from the Cell Ranger 5 and drop. We don't need the first Cell Ranger one because that's redundant because everything that's in the Cell Ranger is in the other ones. And then I'm just going to remove the zeros for the new ones because I didn't do that to the Cell Ranger fits. I only did that for the raws because if you remember, we didn't actually read in the data set from the, I'll scroll back up as I talk. We didn't actually read in the data from the feature barcode. We just read the barcodes. Saves memory, saves RAM, smart thinking, right? So the data is going to be the same. Anyway, going back down. And all we're going to do here now is basically add labels. And this is kind of useful down the road when you're doing your TISNEs and UMAPs. If you see weird clusters or you're kind of trying to figure out stuff, you can actually then graph where they came from. Is this a cluster that only empty drops found? Or is it, and that can kind of let you know where things might be from, or if you should remove them, exclude them, so forth, and further down. So I'm just going to do that. It's always good. Again, you can subset your data unlimited with single cell experiment. You can get anything you want. We're going to quickly do some cleanup stuff. I like edgeR. I've already loaded cowplot. I've duplicated the stuff here. That's fine. Next thing we're going to do is we're just going to change the gene names to row names to the symbol names. So I think if I do this, it'll show me. Yeah, so you can see the row names are the long gene names, right? And that's not going to be very useful for us to explore the data with. We're not going to copy and paste all these into gene card when there's hundreds of them. And we're like, oh, that looks interesting. No. Luckily, with Cell Ranger now, they're actually doing a database query in there. I used to have to do a script that would do the annotation using a database. You don't have to. It's in the data set now. You can just change it, and this one line of code does it. And then now, oh, look, it's gene names. Yay! All right, now I'm going to do a quick annotation. As you can see, I've added annotation to false here. I used to have this true, and it would do this top bit by connecting to an online database and doing it for me. Don't have to anymore, saves time. But I still do connect to some databases for cell cycle and some other QC stuff. This is a custom function. There's nothing in here. I'll run this first. There's nothing in here that you can't do manually. I'm going to open it up so you can see it's all open source commands. I'll go to files. While it's running, I'll just go to this. And you can see it's just the kind of stuff you've seen in tutorials. It's like, hey, look, here's the mitochondrial row names, and I want you to add that back to mitochondrial. Like, there's nothing extra fancy in here, guys. It's stuff that Henry's already done in the previous lessons. I just have it in a custom function so that it just does it all at once. That's all that's happening here. And here's the old code that I don't even run this anymore because like I said, it's all built into Cell Ranger now. But before when it wasn't. And so this is just as cell cycle the same way. These are all built in. These are all functions. Add cell cycle is already a function. It's in single cell experiment for doing your calculating your metrics. It's all part of the single cell experiment package. So you don't have to stress about that. I'll just go to here. And then I'm going to pre-run this. Again, setting a seed. Doesn't matter really what the seed is. Maybe Henry can chime in, but I like to set a seed before every next step I do. And I do that because a lot of other packages reset your RStudio seed within whatever their package is. They might have a random number generator in theirs. They might have a set seed and some built-in functions. And I'm not going to go through every package I run and read all the code of every open source project. So for reproducibility, anytime I'm at a step where I want that reproducibility, I reset my seed. I don't know if that's best practices. I don't know if that's stupid and redundant. It's what I do. Take it what you will. I'll change this to something a little more interesting like the date. And we'll just let that finish running. Shouldn't take too much longer. Any questions? Oh, Henry says, sorry for the confusion. You will know T-cell data libraries needed to run the script. It's just illustration. Yes, okay, you're saying yes. Yes, you won't have the data. run it. Sorry, guys. But that's all right. Hopefully this will be finished up quickly. Is there anything else I want to talk about before we jump ahead, maybe? So one of the things we're going to do once this is done is I'm going to kind of explore the data almost identically to how Henry did it, except I'm going to do it with bioconductor packages. So I'm going to grab the mitochondrial genes. So they're all listed because we've sorted now. We've changed the row names to the gene names. They all have an MT/ in front of them. So I can basically grab all of them by basically grepping anything with an MT. Then I'm going to do the same for ribosomal genes. And I'm going to say, hey, anything that has the RP, that's a ribosomal gene. I want to know what that is. So we're going to grab the mitochondrial, grab the RBs. So we can see now we have a list. These are all our mitochondrial genes. Here's all your ribosomal genes. Some people regress out the mitochondrial genes when they do differential gene expression. You can if you want to. It depends on your experiment, depends on what you're looking at. I don't do it for the RB. I do the RB more because we do myeloma, and myeloma cells die like in flow sorting. They die in cell sorting processes, and so I need to I have more of a cell death problem than I think a lot of people tend to have. Plasma cells are just like that. So one thing I will do is I want to plot the highest expressed genes, but I don't want them to be mitochondrial genes. I don't want them to be death genes. I don't want them to be cell cycle genes. I want to know, are these cells what I expect them to be? So we just do plot highest expression. It's built into single cell experiment as a function, same as those feature plot barcode things are built into scRNA. A lot of built-in graphing stuff here. So that's going to plot itself. I'm going to have to switch back to the plot tab. Hopefully it loads. Well, while that's loading or not loading, I will jump into normalization. Oh, oops, no, it's the old plot. New plot, please. So there's lots of types of normalization. Obviously, Henry didn't go over every kind because he can't. It's impossible. There's not enough time. Okay, so our plot's loaded. It's going to be kind of blurry, but we're kind of looking at this. And can we see the kind of genes that we expect to see this? We're seeing HLA genes. That's good. We're seeing gramzyme genes. So we have cytotoxic cells. Good. We expect that in our T cells. Interleukins, IL-32. So this kind of gives you the idea like, oh, yeah, this is kind of what we expect to see in kind of our cells. So it's kind of like, oh, okay, nothing here shouting out that it's like too crazy. So what we're going to do first is we're going to normalize the library. So it's a type of scaling normalization. So if you think of, you define the library size as the total sum of counts across all genes in each cell. So the expected value is kind of assumed to scale with any cell-specific biases, right? So this is kind of the library size factor if you're reading about this in publications and journals. So each cell then should be directly proportional to its library size, where the proportionality is a constant is such that the mean factor across all cells then is equal to 1. And so this is a step that I don't know for a fact, I assume scRNA does this as part of its normalization step, but it's a first step. So we do that first, done, easy done. I don't run log norm counts normally. I like most people who are using scRNA. I've now transitioned to SC Transform. It's an open source CRAN package. You can download it from CRAN directly. I could import it right now with library SC Transform, and I'd have it. It's not a uniquely scRNA function, but it is built into scRNA now, and I recommend using it if you're using scRNA. But for time, because as Henry said, one of many reasons log normalization is done is because it's quick. Bam. So we're going to do log. Then you can sort of kind of go on and do your normal stuff, run PCA, TISNE, UMAPs and stuff. Not going to do that. You've seen how it's done. It works the same way in single cell experiment as it does in scRNA. You're not going to learn anything different. I will talk about something that maybe Henry's going to talk about today or in the future, which is doublet detection. There's a lot of different techniques for doublets. So when you have two cells in the same gem, the one I've been using recently, and I found to be the most robust for my kind of immunology work, for any other immunologists out there, is doing simulations. So this is basically you randomly take different gems that you have all these gems, right? You randomly take their expression patterns of these gems and you randomly combine them. And then what you're basically creating is this whole bunch of simulated doublets. And then you take those simulated doublets and you compare it to the actual set. And does anything match up to your hypothetical simulated set? Because ideally, the only cells that could be in combination in your experiment are the ones that were in your population. So in this case, it's only sorted T cells. So I don't care about other types of doublet formations. I only care about the possibilities of doublets of different types of T cells. And I've been finding this very useful for my T cells and my B cells and my bone marrows. Bone marrow contains a lot of different cells. I found it useful. There are other techniques out there, some based on clustering methods. So we talked about those UMAPs and t-SNE clusters, and there's some doublet detection methods that are like, hey, this cell's kind of in two clusters, it kind of fits like other clusters, and it compares them like it's probably a doublet, it has features of both clusters. I haven't had a lot of luck. Well, that's a lie. I haven't found that entirely useful for my workflow. But it's something that exists. A lot of people do it. You'll see it in publications. So one of the things you can also look at, obviously, is cell cycle. Again, I'm just using ggplot here. There's nothing really fancy. Oh, wow, this looks so weird zoomed in big. Anyway, as you can see, you might want to look at your cell cycle, see if there's anything crazy going on. Again, you might have an experiment where you're specifically activating a certain type of cell. Maybe you won't see an even proportion, but you know what you were expecting from your experiment. I know I'm looking to expect an equal number of cells in an equal phase. I don't expect them to be senescent. I don't expect them to be hyperactive. This is how I want them to look. If they didn't, this would be a red flag, right? It's part of QC. Some people regress out cell cycle in their analysis. If you read any paper say 2017, 2016, a lot of people regressed out the cell cycle because they were looking at differential gene expression. All those papers were differential gene expression. And they're like, oh, I don't want my cell cycle genes to be influencing my differential gene expression just because they're in a different state of cell cycle. That's not really the gold standard anymore. In fact, cell cycle genes are basically, there's such a small proponent and they'll be so downplayed compared to the types of expression differences, the levels you actually will see in real events. And when you're looking at things at really tight subpopulations, which a lot of us are, because all the big macro stuff's been done, differences in some of these genes are actually important for cell types. So, there's like 20 types of CD8 T cells, right? Activated T cells, naive T cells, exhausted T cells, cytotoxic, memory. Some of the differences are in some of these cell cycle genes. And if I regress them out, I may not be able to have the resolution I need to identify those subtypes. So I keep them in. Most people are keeping them in these days. It's only those older papers that are really regressing them out, or if you have an experimental reason to regress them out. Next thing I always do is kind of make sure the cells are the ones I'm looking at. I use single R right now because it's Aaron Lund's package. Disclaimer, I'm co-writing a book with him on multimodal analysis for site-seq and BCR-seq. So I'm trying to use more and more bioconductor packages because the book is supposed to be able to complete the entire thing with bioconductor. But there are other packages out there. I used to work in Perth, Australia with Al Forrest's group. We actually developed SC Match. Check it out. It's a great alternative. That's exactly the same thing. Python based instead of R based, but otherwise same thing. Again, set my seed, get a reference here. There's lots of databases out there. I put in the comments here, the Human Primary Cell Atlas is another database. I'm using the Blueprint Encode Data database because it's now, as of 2020, it's now 2 databases combined, and one of them is an immunology database. And since I'm immune cells, this makes sense for my data. Use the one that makes sense for your cell types. Then you just run this prediction. Again, really simple. Tell it how many cores you want to do. You give it the reference. You give it your data. It does all the dirty work. I don't know what Henry likes to use. Henry, what do you like to use for cell prediction?
We're actually going to show people how to use single R today, so this is actually perfect.
Oh, okay. I won't spoil that. I won't. You know what? I won't even go into any detail.
No. This is great. They'll hear it twice. They'll hear it twice. And then if I'm doing anything wrong, I'll learn about it and I can adjust stuff before I get there. So I think you know a little bit more about this package than I do.
Fair enough. Fair enough. So then this basically gives you sort of a table here. You can see that, okay, it thinks that 5,000 of the cells are CD4 cells. 5,000 or CD8. That's kind of what I expect. It's got some predictions for some other cells. You can look at the prediction score heat map, and you can kind of take a look at its percent confidence in its in each cell type. As you can see here on the right now, hopefully there's not a delay in screen share. CD8 and CD4s are the vast majorities, with everything else being very, very low chance of what it thinks it could be. Now, for your own fun and your own time at home, when you have more time running this on a server, switch out main, label main, switch this to fine, and you'll be able to go into even deeper details of subsets. You'll get into your memory T cells. You'll get into your cytotoxic C cells. You'll get them all, naive T cells. But that takes longer to run. I'm not going to do it for this tutorial, but it's pretty sexy. So try playing with fine when you have extra time. Keep it at main for tutorials and live demos. And then basically I do everything else you can normally do. This is like everything else you can possibly want to look at. You can look at it in single cell that we've already looked at, that Henry's already showed. But what I want to show right now, if I have time, Henry, I want to show really quickly that anywhere in your pipeline for single cell experiment, you can jump into scRNA. You can do it 50 steps down, or you can do it 2 steps. You can just do that empty droplet thing and then just jump straight into scRNA if you want. It's really, 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 really stupid easy. scRNA likes patchwork, so I'm going to load patchwork instead of cowplot. Basically, I'm going to use create scRNA object, just like we did. If you go to the documentation for scRNA, they have a function called as scRNA, and you can just pass it a single cell experiment object. Don't do that. It's a trap. It's terrible. It's garbage. And what's going to happen is you're going to try to follow a scRNA tutorial for something, and it's going to fail because it's going to be expecting all these little sub metadatas that aren't generated this way, and it's just going to crash, and you're not going to know why. So save yourself a headache, generate it the original way with create scRNA object. Now, all it needs is a count matrix. So we just turn our selected thing into accounts, change it to a matrix from an S4, 1 line of code, bam, T counts, good to go. We now have a matrix of counts. The only thing that this doesn't have that scRNA is expecting is a bunch of cell names along the top of that count matrix. So we have our barcodes in our single cell object already. Bam, give those names some barcodes, done. Then we just say, hey, create that object. And it's going to do the thing. It's going to do its thing. Anyone here watch Avatar? Do the thing, Jolie. It's going to do it. It's going to be fine. Then you can do everything the normal way. This is kind of an old way. If you guys are ever interested in how scRNA, like version 2 used to work, this is how you did your ribosomal and mitochondrial stuff. But since 3 and 4, this is the new way, where you could just get percent feature set, and then whatever pattern you gave it, it would generate the math and do it as a percentage of the total. Before, you had to like grab your gene names and calculate a percentage of the whole and add that back as metadata. Add metadata still works, and this is actually how I add all my site-seq and BCR data to my scRNA objects. But for this kind of stuff, you don't need to do that anymore. It'll calculate the percentages for you. And then you can create your violin plots like we did before. You can normalize your data, find your features, get your variable features, look at different cell types, subset your data, be like, oh, look, I only want this many features. I don't want this percent of mitochondrial DNA. Do your transform. If you weren't passing transform data, by default counts will just take the other one. But if you wanted to pass, let's say you already did SC transform, you can pass the other array matrix. It's just stored in a separate array. Yeah, and so that's pretty much kind of it. I was gonna kind of show this thing, but it seems kind of redundant now. So that's what I got for you guys. If you have any questions, shoot them in the chat. I'll answer them. I talk fast, but I'm also trying to be aware of the time because we always go over time, and I've taken up a lot of time.
Okay, that was awesome. Thank you so much for giving that. Yeah, I honestly, I learned so much just watching you go through this. So this was really, really cool. And I had no idea that the as.swearout function is a trap, because like I kind of assumed since as.single cell experiment works that it would work the other way, but I'm now thinking maybe it doesn't. Okay, so does anybody have any questions for Wes before we switch over to the main lecture? I'm gonna be curious, Wes, when we get to the part where I'm trying to pretend like an immunologist and interpret the PBMC data. I'm gonna be curious to hear your thoughts on some of the markers we're picking and how we're like biologically interpreting it. So That would be
Sounds
really
good.
nice once we get there. Yeah.
All
Okay.
right, take it away. I'm sure if there's questions, email them. Catch you guys.
All right. Awesome. So virtual round of applause for Wes. That was really awesome. Thank you so much for giving that lecture. All right. Let me go ahead and share my screen now. Okay. Okay. So it's very likely that we'll go over time. So again, the lecture and everything's online. So if you do need to leave early, feel free to do that. But I want to make sure we get through everything because we're going to be talking about what is one of the most difficult and important parts of all single cell analysis, which is biological interpretation. And in particular, how do you assign cell types and how do you understand the biological significance of your clusters? It's really how you tie the computational side of things back to the initial biological questions that you had. Okay, so it's very important for us to understand how that works. All right, so what have we done so far? Well, we previously talked about how to load the data, do quality control, and normalize and scale it. Wes mentioned scTransform, which I actually personally use. I just didn't use it in this workshop series because it is slower and it can cause some other issues when you try to use functions that want you to have log transform counts. But in general, it's the one I would personally use. So he showed what that is. It was the scTransform function in swerat. So it's a great alternative. I'll probably put it in the script for next lecture. We then talked about the PCA and nearest neighbor graphs, and we talked about clustering and low dimensional embedding. So we showed you how to do UMaps, and we showed you how to do your Louvain clustering. Okay, so now it's time to start moving on to advanced topics. It was really nice because Wes actually showed you one of the things that we were going to talk about, which is cell cycle analysis, which is basically where you figure out which cells are in which phase of the cell cycle based on what they express. But instead, what we're going to focus on today is mostly going to be cluster marker analysis and cell type identification. And then hopefully next time we'll have time to get to all of subclustering trajectories and some more cluster DGE. Okay, so let's first talk about cell type assignment. So the goal of cell type assignment is to take you from the point where you have here, you can see these unlabeled clusters. So we know that these clusters are somehow biologically significant, right? Because we uncover them in an unbiased way from our gene expression data. But we want to figure out now what cells do they correspond to. So we know this is a PBMC data set. So after doing our cell type analysis, we should be able to figure out for each cluster what kind of cell is it. So we're seeing B cells here, NK cells, and a whole other bunch of stuff here. Cell marker or cluster marker identification is all about, instead of trying to figure out what is the cell type label for something, is trying to figure out what are the pathways and biological processes that are overrepresented in that cellular population. So for example, if we focus here on cluster 0 and we do our cluster marker identification, then we do an enrichment, we can identify what are the biologically significant pathways in gene sets which are overrepresented in that group of cells. So basically, I know what is it that these cells express in terms of genes, and I know what biological processes those genes relate to. Therefore, I know what biological processes are overrepresented in that cellular subpopulation. This is something that's not going to be super important today, but it's going to become extremely important once we start getting into identifying things like tumor cellular subtypes and metastatic subpopulations and hypoxic subpopulations in tumors. And, same thing in Alzheimer's disease, finding dead and dying neurons or neurons that have somehow survived, tau and amyloid beta. So it becomes such an important part of your ability to interpret your data. And that's it. Let's jump into the R now. Okay, so hopefully you have already got module number 4 pulled up in front of you. If you don't, this is now your chance. So go on to GitHub. I put the link in the chat. I'm going to put it there one more time. Go on to GitHub. make sure you have the latest version of this. So you can download the zip folder here and then just double click on that R project file, and that will take you right here. Okay. So make sure that everyone, I want to make sure everyone is actually here. While we're waiting for anyone who's catching up, go ahead and run these first few lines, 5 through 27. So basically what this is going to do is it's going to check for any missing packages from the normal CRAN, like these are the normal R packages here. So CRAN is the repository of most R packages that exist. And you're also going to check for packages that are from the bioconductor repository. So these are different repositories, and the way you install them is actually different. So with CRAN, you can do install.packages. With bioconductor, you do biocmanager install. Either way, this is going to check to make sure you have them, and if you don't, it's going to install them. Okay, so I'm going to give just a second for people to do that. Yeah, if anyone has any questions, do put them in the chat. I want to make sure we get to all your questions because this part's going to be pretty difficult compared to what we've done so far. The coding is a bit more difficult, and the actual conceptual nature of it, depending on what your background is, will be more difficult. This is the part where it really is important to have biological knowledge yourself or to be working very closely with someone else in your lab who is a biologist. Okay. So hopefully you've gotten all of these packages now. All right. So go ahead and load them up. Make sure you've got all of them loaded. And we're going to just recap what we talked about last time. So as we showed before, you can chain together your commands in scRNA. Okay, so you can see this here. This is a long chain of commands. And in fact, this chain of commands here covers everything that we've already done to this data set. So let's go get that PBMC data set again, untar it. And now what we're going to do is this first line, it's going to read it into a scRNA object. We're going to do the percent feature set. We're going to subset it. So this is our QC. Normalize, variable features, scale, PCA, find neighbors, build your nearest neighbor graph, find clusters, that's low vein clustering, and then you're going to do your 2D embedding. So what it took us 3 weeks to learn, we actually can accomplish in what is less than 10 lines of R code. In fact, depending on how you think about it, it's actually just 1 line of R code. Pretty cool. So you can put your cursor anywhere here and hit Control Enter or Command Enter, and it will just run the whole thing. And there it goes. Pretty fast. And we're done. All right. There you go. 3 weeks in about 10 seconds. All right, so let's just recap again. Where did we end up last time? Well, we have our UMAP and our clustering. So let's go ahead and plot that. So that's line 53. Okay, so this should look pretty familiar. This is our PBMC data set with all of our clusters in a UMAP. Okay. So what do we want to do? Well, we want to start to understand what is the biological significance of each of these clusters. So let's go ahead and pick one. Let's pick cluster 3. We want to now figure out what are the genes that are expressed specifically in cluster 3. Or we could say it another way. We could say, what are the genes that are overexpressed in the cells of cluster 3? And that might give us a clue as to what are the cell types in that cluster. What does that cluster represent in terms of the biology? Okay, so how do we do that? Well, in scRNA, it's easy. You can just use the find markers command. So we're going to call find markers. We're going to say PBMC because that's the name of our scRNA objects. We're going to say ident.1 equals 3. So we're saying we want to find cluster 3's markers. Only positive equals true. This basically means we're only going to find the genes that are overexpressed in cluster 3 compared to everything else, as opposed to underexpressed. We're going to set a log fold change threshold to 0.5. This is just a way to speed up the computation time. We're basically saying we only want to return results when the average log fold change of the expression between cluster 3 and everything else is at least 0.5, log 2 fold change of at least 0.5. And then min PCT is basically the minimum percentage of cells that that gene has to be expressed in for it to be returned. So because we're doing only positive is true, min PCT only applies to cluster 3. So all the genes we return are in at least 25% of the cells in cluster 3. Okay, let's run it. Done. If we take a look here at the resulting data frame, we can see here it's sorted in order of p-value adjusted. So this is the p-adjusted value. And we can see on the row names the genes that it's pulled up. So MS4A, CD79A. One of the things that you want to keep in mind when you're doing this sort of analysis is that the p-values can be just a bit overinflated, okay? So it's really helpful to actually just focus at first when you're going through the data in the first pass to just focus on the very top results. So let's go ahead and filter this, and we're just going to take the top 5 results based on the p-value. All right, so now we see MS4A1, CD79A, CD79B, a link, RNA of some sort, TCL1A. Okay, so anyone not Wes, can you tell me what is the likeliest cell type identified here? What do you think cluster 3 might be? Just take a guess. And you can put it in the chat if you want. Okay, so I see NK, I see B. Okay, anyone else want to chime in? Okay, I see another B. All right, so it is B. It is B cells. Yeah, more Bs. All right, so B cells. So how did we know that? Well, let's take our very first marker. All right, actually, let's take our top 2 markers. We'll take MS4A and CD79A. First of all, when we do a feature plot for these, we can see that they are quite specific to that cluster 3, okay? Right, so they're quite specific, which means they are probably specific markers of a cell type. If I take MS4A and I just simply Google MS4A gene, which apparently I've done before, even before I get to the gene cards page, I can already see B lymphocyte antigen CD20. But let's just say I got here. Okay, so basically I'm reading here uniprot B lymphocyte specific membrane protein. And I think CD79A and B are actually the B cell receptor, if I'm correct. Wes, anytime I say something about immunology, do feel free to correct me if I'm wrong, just as a general rule. Okay, because immunology is really not my background. All right, if we look at our violin plot, we can actually see this another way. And in this case, instead of looking at all the cells kind of at once in a UMAP, we're now directly comparing the clusters for their expression of these markers. So we can see cluster 3, which is our presumptive B cell cluster, very strongly expresses this. So for those of you who don't know, a violin plot is basically like fancy box plot, right? It's like box plot, except it's not just a box. You can actually see the distribution, but it kind of functions in the same way. So you can see here that CD79A and MS4A1 are pretty specific to cluster 3. This helps us see that cluster 3 is probably a B cell cluster. But as we're going to see, there's lots of other ways to determine this. Come back to our UMAP, we might notice something else, which is that you have what appears to be a group of clusters. So these are all clusters that if you weren't looking at the UMAP, you wouldn't know if they related to each other in any way at all. But because you're looking at the UMAP, you can actually see they seem to have some sort of relationship in our graph, right? So the cells on the graph seem to be, for some reason, connected to each other. And this gives us the idea that when we're looking at single cell data, it's very helpful for interpretation purposes to see both the embedding and the clusters in one visualization, because they can each tell you things that the other can't, okay? We might want to ask the question now, well, all right, so clearly there's something about these 4 clusters, 0, 2, 4, and 6, that is shared. They have something in common that makes them different from everything else in the embedding, all the other clusters. So let's see if we can now figure out what are the genes that are expressed in all 4 of these clusters compared to everything else. So how do we do that? Well, we can actually, for fine markers, we can actually add multiple clusters at once. And so you can see here, we can specify clusters 0, 2, 4, and 6 all together as ident.1. Okay, so we go ahead and run that. And now let's take a look at our top 5. So we can see here, very significant, IL-32, CD3D, CD3E, MALAT1, LDHB. Okay, so let's go ahead and plot the first two, IL-32 and CD3D. Okay, so something interesting here already is taking shape. We can see that IL-32 seems to light up in all the cells in that group. But CD3D only seems to light up in part of it, but perhaps it's missing from one of the clusters. We can confirm this a little bit more closely with the violin plot. Okay, so again, IL-32 clusters 0, 2, 4, and 6 seem to all overexpress that, perhaps even a little bit cluster 7. But CD3D is only in 0, 2, and 4. 6 is quite conspicuously missing that one. So this is now going to be a much harder series of questions. So I tried to softball you for the first one, but can anyone tell me what CD3D is a marker of? Can anyone tell me what CD3D is a marker of? Okay, so I'm seeing two T's. Okay, good. Yeah, T. Yeah, CD3D is a T cell marker. Again, how do I know this? I can come here, Google it, and I can already see, even before I jump to gene cards, it says T cell surface glycoprotein, T cell surface, T cell receptor. Yeah, so T cell receptor. So it's actually part of the T cell receptor complex. So very strong marker of T cells. So what is that implying then? Well, it might be implying that there is some cell types, including T cells and something else, that all share expression of IL-32, but it might be that this cluster here, cluster 6, is actually not a T cell, but something similar. Or perhaps it's a T cell, but one that is negative for CD3D. There's both possibilities here. Okay, so last question. Can anyone tell me what IL-32 might be a marker of? Or can anyone take a guess or a quick Google as to what IL-32 might be a marker of? Okay, that was pretty fast. This was supposed to be a much harder question, so you guys are very impressive. So okay, so let's I'm gonna slowly move to my browser. Well, if anyone else wants to chime in while I'm doing this. Okay, so I'm seeing a couple people saying NK. Okay, I'm seeing quite a few people saying NK. All right, so if I read the description for IL-32 gene, I can see that, in fact, it seems to be, in some cases, expressed by T cells and perhaps even NK cells. So, okay, very good. T cells and NK cells seem to be able to express this. And that actually fits with what I'm seeing here, because it can't just be a marker of NK cells, or else the T cells wouldn't also be expressing it. So it seems to be potentially a marker of T cells and NK cells. And maybe that's implying cluster 6 is NK cells. It could also be, so, alternative hypothesis, right? It could also be that CD3D is just simply not expressed on some T cells. And that actually is true. There are some T cells that don't have CD3D. Okay, so it could be NK cells, but there's an alternative hypothesis we also have to consider, which is why we don't use just one approach to figuring out cell type. And in fact, I would argue what we're currently doing is probably the slowest and least effective of all those approaches. So Wes made an interesting point. He says CD3 is also on some B cells. I actually had no idea. I feel my knowledge of immunology is quite weak, so it's very nice to have that info. Okay, so what I want you guys to do now is I want you to go ahead and do this activity. So this is going to be to basically redo what we just did, except with a brain data set that comes from what's called the tabula muris. Okay, so for those of you who haven't heard of tabula muris before, it was a landmark single-cell RNA-seq paper from 2018. I put the link in the actual script. And basically what they did was they took 20 mouse organs. They did both kind of droplet-based single cell, like 10x genomics kind of single cell. They also did fax-based single cell that was full transcript. So they used something called smart-seq for that. So it was really high quality data, really fantastic data set. And what I did was I took the brain fax data from it, and I've already given you the a small version. So I cut out some cells, a small version of that data set that's only got 2,400 cells. And I've already set up the pipeline here for you with the number of dimensions selected and the quality cutoff selected. So go ahead, run these. It's kind of slow because it's very highly compressed to fit under 10 megabytes. It shouldn't take terribly long. Okay, and then go ahead and run this. Just takes a second. Found 8 communities, which is great. All right, and now we can go ahead and take a look at the UMAP. So this is your starting point. What I want you to do is to find the markers for cluster 0. I want you to do a feature plot on the top 2 markers from this cluster. I want you to do a violin plot, and I want you to take a stab at figuring out what kind of cells are in this. Bearing in mind that these are brain non-myeloid cells. So these are non-myeloid cells from the brain. I actually don't think there's really any immune cells in this data set. So these will include things like glia and neurons for the most part, and endothelial cells and pericytes and that sort of thing. But I want you to go ahead and take a stab at that. I'm going to give 5 minutes to do this. So let's go ahead and regroup at 6:10. And if you run into questions, do feel free to put those in the chat. Okay, so I've gotten a few questions. I'm just going to quickly address them, and then we'll jump into the answer for this. Okay. Oops. All right. Okay, so first question is how are we supposed to be installing these packages? So right. Okay, so I just wanted to double check this, make sure this all made sense. But yeah, okay, so what you're doing is you're running these lines, and what it's going to do is if you don't have it on your computer, it should install it here. So has anyone not been able to install CellDex by running these lines? CellDex you cannot install with install.packages. It's only a bioconductor package. You have to use BioC Manager install. Has anyone not been able to accomplish that? Because you will need CellDex for this.
Quick question for you, Henry.
Sure.
Which database are you going to be using from CellDex?
It's I think it's like called Monaco or something like that. Monaco Immune Cells, I think.
Okay, cool.
Yeah.
Because CellDex in the newest version of SingleR is mostly redundant for most databases. They're built into SingleR now.
Oh, okay. Okay, but I think SingleR has to call CellDex, doesn't it?
No, it has a built-in function to do that. You don't need to install it separately or call it separately.
All right, so yeah, never mind. You actually apparently will not need CellDex.
But there are some databases in CellDex that aren't in SingleR, so I wasn't sure. It depends on which database you're using.
We're also going to use the mouse RNA-Seq. So hopefully that one's also, but yeah, you should have been able to install that with the BioC Manager install command. So just to like recap, that's BioC Manager install CellDex. Right. So it says it's already in use and will not be installed, and that's because I already am using it. So if you get anything other than what I just got when you try to do that, put the exact text of what you got into the chat. Because if there's an error, then we can help you, but we have to know what the error is first. So it's kind of just a general rule of troubleshooting. Put the text of the error into the chat. Okay, so apparently even if you're having trouble doing that, you can use the remotes install GitHub way. So yeah, so Ramesh just put that in the chat. So that would be an alternative way to do it. Shouldn't have to do that, but if you are for some reason not able to get bioconductor to work, that's another way. Okay, so now we're going to answer these questions. Okay, so like so many things, you should really just copy and paste when you can. So let's go ahead and do that here. All right. So we're going to change out PBMC for brain. Now, stuff you do want to change. So we're going to be looking for cluster 0, as you'll recall. So let's look for cluster 0. So cluster 0. So, okay, maybe that required a little bit more work than it would have required just copying and pasting. But now we can see what our top markers are. Okay, so let's do a feature plot for our top markers. So we've got MOG. And so this is mouse data. So the gene names will be has a different style. The first letter is always capitalized, and then the rest of them are lowercase. So we'll copy and paste these here. So let's go ahead and do our feature plot now. All right, great. So this pretty strongly highlights that cluster 0. Pretty strongly highlights cluster 0. So this is a strong marker of cluster 0. So all this stuff was actually extraneous. The last question was, what cell type is cluster 0? Okay, so anyone had a chance to look this up? What cell type is cluster 0? So feel free to put in the chat. Okay, good, good. All good endocytes, good, very good. It looks like people got that. So again, how did I know that? If I just take the very first one, MOG, and I put this here, MOG gene, very first thing that comes up is MOG. The O in MOG stands for oligodendrocyte. And if I read about it here, I can see it's really an oligodendrocyte, a relatively specific protein for oligodendrocytes. So if I go through the top 5, I'll continuously find that. So great. So it was oligos. All right. Any questions about that before we move on? Okay, well, if any questions arise, do put them in the chat and I will make sure to get to them as soon as I can. All right, so that's one way to identify cell types. But it tends to be very slow, very inefficient, involves a lot of Googling. And sometimes the top markers for a cluster are not clear cell type markers. Okay, so what do you do when that happens? Well, another way to approach the problem that is synergistic with that first method is to actually start from a list of markers that are pre-identified from the literature or from various databases and resources online. So this particular marker list, I actually just stole it from the PBMC tutorial from scRNA-Seq, but I'm pretty sure they stole it from the literature. So that's one way you can do it. So this came from, let's see here. Where did that come from? Okay. So that came from right here. All right. But don't look at it because that'll tell you what the clusters are. It should be a surprise. All right. So basically we said naive CD4 positive T cells are these. Memory CD4 positive T cells are marked by these. I don't know how accurate any of this is. If you were an immunologist, you would take the time to go painstakingly through the literature and find the correct markers that you can support in whatever publication you end up going forward with. But for a first pass analysis of your data, it's fine to just use a panel that you found somewhere online, because you don't yet have to justify what you're doing to someone that you're trying to get to publish your paper. So this is just about exploratory data analysis at first. When you're ready to go back and really properly find everything, then that would be when you go back to the literature and you find the studies that show what the markers are. And actually, we'll see in a minute how you can sort of do that in a semi-automated fashion. But for right now, let's pretend like we are starting from an a priori marker list. A priori because it is prior information that we know ahead of time. All right, so how can I use this prior information list to identify the various PBMC populations which I think should exist in my data? So let's start with B cells. B cells was something that we've already kind of identified before, and we know MS4A1 and CD79A are B cell markers. So if we do a feature plot with those B cell markers from the marker list, so this is how you can access those B cell markers. If I just print this to the screen, we can see that's just the vector containing those two genes. We can see, again, pretty strongly identifies our cluster. And if we do a violin plot, if it finishes, we can see, again, pretty strongly identifies our cluster of interest. So cluster 3. And we can go all the way through. We can do CD4, we can do CD16 monocytes, we can do NK cells. So remember when we thought that that cluster 6 might be NK cells? Let's go ahead and take a look now. Okay, so does seem that these NK cell markers might be pretty specific to that cluster. Do the violin plot. Okay, again, relatively specific to cluster 6, but as you can see, cluster 4 also seems to express these to some degree. So this is where you start getting into why this is actually the hardest part of single cell, because you will find very quickly there is uncertainty. And unless you've got the biological knowledge backing up what you're doing on the computational side, you're going to have a really hard time interpreting your data. Okay, so you could go run all of these one by one, but instead I just made a function that's going to take a scRNA object, a marker list, and a marker that you currently want to examine, and then it's going to make that violin plot. And then I just apply that function to all of my marker list markers to make a list of violin plots for every marker. And then I do this GG arrange to compile it all into one plot, and then I can just save the plot. And when you do this, you can go back to your module 4 here, and you should see that it's shown up right here as a PNG file in your folder that you were in. Okay, if you double click on this, it now shows displayed very nicely for you all of the cell type markers and the the cell types and the markers that you had from prior knowledge. And you can go through these by hand now, and you can scan and you can say, okay, are there any of these that seem to clearly identify a cluster? So in looking at this, are there any of these besides the B cells that seem to clearly identify one specific cluster? And you can pick any, you can just shout it out. And Wes, do feel free to chime in if from like the immunology perspective, you have some thoughts on why this is like not ideal, because again, like that is not my background. So if you're an immunologist, you might think of this differently. All right. So anyways, so if I'm looking at this, the first thing I'm going to notice is, sure, my B cells pretty strongly identify, my B cell markers pretty strongly identify one cluster, that's cluster 3. But if I look at my CD8s, T cell markers, they also seem to pretty strongly identify one cluster, that's cluster 4. Same thing with my dendritic cell marker FCER1A, but CST3, which is also supposed to be a dendritic cell marker, is less certain. It's much more ambiguous, but this one seems to be pretty direct. All right, finally, PPBP, which is my platelet marker, pretty clearly in cluster 8. So that one I'm feeling pretty solid about. So this is another way to identify the cell types and see which clusters belong to which cell types. But as you can see, there's actually quite a bit of ambiguity. So this is complementary to what we were doing earlier, but again, neither approach alone gives sufficient information to be able to say this cluster is this cell type, unless it's just super clear cut like B cells. Okay, so go ahead, take 3 minutes now, and what I want you to do is do the same thing, but with the brain data set. Okay, so I want you to do this exact same thing we just did. The function, you can literally use this function, but I want you to use this VLN list. I want you to save it as a plot. I want you to save the plot, and then I want you to look at it and see if there are any any cell types that you can very clearly identify just by looking at that plot. So go ahead, take 3 minutes. We'll come back at 6:26 or 6:27. Again, if you have questions, feel free to put them in the chat. Oh, and I totally forgot to do this. The code from the last activity, I'm just about to post that in the chat. Sorry about that. So this would be code from activity 1. So my answer to activity 1, essentially. Okay, so hopefully that all worked for you guys. Mauricio says he didn't see the PNG file, but it will be right here. If you're using an R project like you should be, it will be right here. Okay, unless you put some other random file path in here, it will always be in your working directory, which is where your R project file lives. Okay, or that's, considering whether, if your GG save had an error, then it won't be there, but it will tell you if it has an error typically. So you can even, I think you can even open this. Well, I guess you can't open it directly with RStudio, but you'll at least see it when you come here to the folder that you're currently working out of. And you also ask, what is the code for what kind of cells are likely in this cluster? There was no code for that. It was just oligodendrocytes. So there was no code for that part. That was just the oligodendrocytes. Okay, so let's see here. Okay, all right. So I want to ask if anyone is willing to volunteer and show their analysis and explain which ones they felt were easy to assign. Okay, so if anyone wants to volunteer, I know it's like a big ask, but I'm very happy to share the stage here. Okay, so I'm going to go ahead and just show how I would do this then. All right, so what I'm going to do is I'm going to, okay, that's right, I'm not showing my screen, am I? Keep forgetting that. All right, so what I'm going to do is I'm going to, because I'm right here now. Okay, so what I'm going to do is I'm going to take this code, I'm going to copy it, paste it, and then I'm going to do a find and replace PBMC with brain. Done. The other thing that needs to happen, of course, is the marker list has to be changed over because now it's marker list brain. But that's really the only other thing that has to be modified. All right, so go ahead, run that. Should be done. GG save. And then let's go ahead and take a look at that. So like I said, if you're using our projects like you should be, this will show right up, show up right in your working directory, as long as you didn't get an error. And there it is. All right, so this one's a little bit more complicated because we had more markers. So the other one had 2 per each, and this one has mostly 3 per each cell type. So one of the nice things, if we come over here, we can see that at least with the oligodendrocytes, as we pointed out earlier, they're in cluster 0 probably. And cluster 0 is pretty specific for the oligodendrocyte markers as well. So cluster 0 seems to be pretty strong oligodendrocyte identity. So we would probably feel pretty comfortable in saying that's an oligodendrocyte cluster. Oligodendrocyte progenitor cells seem to be in cluster 4 for the most part. Okay. Endothelial cells seem to be in cluster 1. So these are pretty specific to cluster 1. Neurons seem to mostly be in clusters 5 and potentially also 7, but we'll come back to that later. This is a little bit more confusing, a little bit more ambiguous. Astrocytes seem to be mostly cluster 2, although again, there's some ambiguity here as well. Pericytes seem to be mostly in cluster 6. So these are both relatively specific markers of pericytes. So I feel relatively confident about this, but we'll have to get back to it in the final step. And then I don't think we had any neuroepithelial cells. So hopefully this is making sense. You can use prior knowledge in a faster and more efficient way than just guessing and checking from the cluster markers. But there are downsides. There is inherent ambiguity sometimes, and it all depends on the strength and robustness of your prior knowledge. So again, here, ton of ambiguity. And I didn't build this list myself, so I have no idea how much research someone put into identifying these markers. They might honestly not be great markers. Who knows? Or they might just be ambiguous. So you have to deal with that fact that most of the time it's not going to be cut and dry. I've tried to give you a little bit easier data set here because I want you to be able to do this in a timely manner during the workshop, but just recognize that if this feels frustrating when you're actually doing it, it's because it is. One way that we can help, though, is by doing our final approach, which is automated classification using CellDex and single R. Although we've actually learned now that you actually don't need to have CellDex. You can do this all with just single R. So we go ahead and load up this Monaco immune data set. And so the way that single R is working is you have reference data sets that contain some cell types that you're interested in classifying, right? Okay, so in this case, we're dealing with mostly immune cell types. So if I take a look at this, we can see it's a class summarized experiment. So summarized experiments are often a bioconductor class structure, which is used for containing data sets that contain some sort of genomic data. Typically, like RNA sequencing data is contained in these. And it'll have, as far as the features are concerned, you can see that actually is quite similar to scRNA-Seq object, but the main difference is that the things we're going to want to look for are not just the counts, which are in assays, just like they are in scRNA-Seq, but also the call data. In scRNA-Seq, that's called metadata. In summarized experiments, it's called call data. So call data is the object which is describing your samples typically. So just so you know what that is. Now, if we take a look here at our large summarized experiment, we can see the call data here. We can expand it, and I'll just show you a trick here because it says it's a D frame, but it's actually, you can get it as a data frame. So if we do, yeah, we viewed it. If we do view mimd.se call data, and we'll make this as.data.frame, we can see it as a data frame. So we can see each of our samples is the row names, and then we have 3 labels. So we have a main label, which is what Wes used earlier. So that's kind of a larger cell type label. So we know for this sample that it happens to be CD8 T cells. We have a fine grain label. So we're saying now more specifically, they're not just CD8 T cells, they're also effector memory CD8 T cells. And then you can use the ontology label. So this comes from the, I guess, the cell ontology. I don't typically use these. A lot of people do, but I just don't see any reason to do it, at least in a first pass of the data, unless there's something very fine grained you're trying to find. Typically, these are totally sufficient. Okay, so these are call data, which means they're describing columns. Columns of what, though? Well, they're describing the columns of the assay, which is the log counts. So if I just take a quick look at this, I can see here that it is a matrix containing a lot of samples. Looks like, yeah, 114 samples. And it's containing, on the rows are genes, on the columns are samples, and then in every cell we have the log counts for this sample of this gene, right? So this is just a count matrix, except it's log transformed. And so the reason we use this is because this comes from a bulk RNA-seq data set. So it is inherently more robust than a single cell RNA-seq data set. And because we know very clearly which cell is which, we can say now, okay, expression of this gene is relevant to this cell type. And by examining this and comparing it to our data, this is labeled, our data is unlabeled, we can transfer the labels from the reference data set to our unlabeled data set and therefore label it. Okay, so this is very machine learning heavy. I'm not going to explain how it works, but just know that to do it, all you have to do is first turn your scRNA-Seq object into a single cell experiment. So that's what we're doing here, using the as.single cell experiment function. And then all you have to do is give your single cell experiment as the test argument to single R. You tell it what the reference is. So our reference is mimd.se. And then the labels, we want to use the main labels. So instead of the find labels, we're going to use these main labels. So we just give that as a vector like this, and then run it. So it typically just takes a minute. It's going to take for, okay, so it didn't take that long. But for every single cell in your SCEP, which is your swear out object, it's going to try to find the highest confidence label from the reference set that can describe that cell. So I'm pretty sure every single cell is going to get a label. It's just a question of whether or not it's accurate that ends up being more contentious. So one of the ways that you can see your results is actually to plot them as a heat map, which actually Wes showed earlier. So let's go ahead and now do that. Okay, great. So I have sorted these by clusters. Okay, so Ramesh got a very weird error, and that is not good. Let's see here. All right, you have a different version of bioconductor than I do. Okay, so you know what the error is. All right, yeah. I know you're on a different version of bioconductor. I bet updating bioconductor will fix it, but if you know a different fix, Wes, can you, just for anyone who watches the video later, can you also either unmute and say what the fix is, or you can put it in the chat and I'll just read it out. Because yeah, I know like errors like that are just super frustrating because it's like, it appears to be something wrong with the person's with the package itself. So it can be really annoying. So the error that, just for people watching the video later, the error that Ramesh got is, it says error in .local. Here, I'm just gonna show this here. Error in .local, xij equals j, I must be length 1. And he got this when trying to get the CellDex data. So the reason I was saying that he has an older version of bioconductor is because when I run this function, it says snapshot date is 10/27, which means that we're probably on different versions of bioconductor. But yeah, apparently the fix is to restart your R session. So sorry about that. I know that's super annoying, but hey, you got a fix though. Okay, so let's take a look at this now. So just like Wes was showing earlier, you have the clusters, and now you have the, for every cell, the degree to which that cell is described by a particular label from your list of labels. So we can see for cluster 0, it seems to be pretty well described by the label CD4 T cells, and then a little bit less CD8, and then a little bit less just normal bland T cells. Okay, and then we can see cluster 1 seems to be more monocytes, and you can go through all these by hand. But what's nice is that single R has actually already assigned the labels for each cell. So you don't have to do it by hand. You can just, this is a way to assess them, but it's already done it for you. And you can see them at the top. So that's the broad labels. Now let's do the fine grain labels. So the only difference between the fine grain and the broad labels is that we select the label.fine column. So we selected the, okay, there it is, label.fine column here. Go ahead and do that. Same thing. It shouldn't take terribly long because it's a small number of cells, or at least relatively small number. All right, there you go. Okay, so let's go ahead and plot the results now. Okay, so this one's a little bit more complicated because, again, we now have our fine grain labels. But still, you can see some pretty interesting assignments of cell type for each cluster. One of the things you might notice when you're looking at this and when you're comparing the broad and the fine grain labels is that there are some pretty distinct advantages to taking one versus the other. And they actually are synergistic, because the disadvantage of the broad labels is that you are losing out on granular information. But the advantage is that you have a much more confident way to map the labels to your data because they're broader labels. More things can apply. The fine-grained labels, you lose some of that power, but you gain the ability to see more specific cell types. Okay, so there's a trade-off to be made. And one of the ways that you can get the best of both worlds is by doing both and then combining them. So I found this function. I really just started using single R relatively recently. I don't typically do my cell type analysis like this because I'm often dealing in cancers where it's not quite as relevant to try to find, different like immune cell types and that sort of thing. But I started trying this package out, and this was actually one of the functions that most stuck out to me, that you can take your results from the broad analysis and the fine grain analysis and actually combine them together and have it recompute and get the best of both, essentially. Or at least that's in theory how it works. And in practice, I've also found it to work that way. So it spits out this warning, which I don't think matters. Someone correct me if I'm wrong, and Wes correct me if I'm wrong, but I don't think that warning really matters much because it seems to still work fine. All right, and if you plot the heat map, you can now see all three together. You can see your fine grain scores, your broad scores, and now your combined scores. And you can see the cell types associated with each. So cluster 0, we now say they are CD4 positive T cells. Cluster 1, we're saying those are monocytes. Cluster 2, CD4 positive T cells. B cells for cluster 3, so that recapitulates what we thought it should be. Cluster 4, it's saying T cells, which is really interesting because we were having a hard time figuring out how those were different from NK cells earlier, which it says now are cluster 6. So that basically recapitulates what we thought it should be. And dendritic cells it found in cluster, I don't know, I guess that's like 7. Yeah, cluster 7. And then it says progenitors are in cluster 8. And we'll get into a second why it's saying that and what that means. So again, all these ways of finding cell types, they all have flaws and downfalls, which is why you have to combine them and use your intuition and use your knowledge of the biology to interpret them. So one of the things we can do is we can take the labels that we discovered, add them back to our scRNA objects, and then we can compare the clusters to the labels. So this table command is going to take 2 columns, the predicted ID column, which we just made with our labels that we got from single R, and it's going to take the scRNA clusters. Now it's going to compare them to see what labels were applied to what clusters, and then how many cells for that cluster had that label. Okay. So go ahead and do that, and you should be met with a print that looks kind of like this. Okay, so across the top are your clusters, across the side are your labels, and then each value is the number of cells in that cluster that got assigned that label. All right, so cluster 3, we know these are probably B cells, and we can see that B cells are by far the most common label. There's a couple that seem to be outliers, but we're pretty confident now in saying cluster 3, definitely B cells. And there's a few others here that are kind of interesting to look at, but there's also clusters that have some ambiguity in them. So cluster 0 seems to have 480 CD4 positive T cells, 157 CD8 positive T cells, and then just T cells in general here. So there's some ambiguity here. How can we deal with this? Well, one of the ways is to just go back to our plot from earlier from our prior biological information. So going back to our, whoops, that's not what I was looking for. Going back to our arranged PBMC violins. And then let's take a look at cluster 0 with respect to the CD4 and CD8 markers. So it does have those CD4 markers. Okay, CD4 markers. And it doesn't seem to have the CD8 markers that we would have associated with it, such as CD8A and GZMK. So even though single R has kind of created some ambiguity here, and that could be due to issues in the references, it could be due to a lot of things, we were pretty confident that despite all that, these are probably CD4 positive T cells. So if we wanted to really verify this, we could go through it with a finer tooth comb. But since this is just a first pass analysis, I'm pretty comfortable saying cluster 0, CD4 positive T cells. Okay. So let's do one more. So clusters 1 and 5 are kind of interesting because we said from single R that they are both monocytes, but they're in different clusters, which means there's something different about them. If we look back at our prior knowledge graph, we can see here that we have two potential subtypes of monocytes that we have prior knowledge about: CD16 positive monocytes and CD14 positive monocytes. Interestingly, if we look at the markers for CD16 positive monocytes, including CD16, we can see that they actually both light up cluster 5. The CD14 monocyte markers, at least CD14 does not light up in cluster 5, although there's a little bit of ambiguity because of YZ, but CD14 seems to light up in cluster 1, whereas CD16 does not light up in cluster 1. Okay, so when I come back here and I say, single R told me that cluster 1 and cluster 5 are both monocytes, well, I'm actually pretty sure that, in fact, cluster 1, they are monocytes, but more specifically, they're probably our CD14 monocytes, and cluster 5 are probably our CD16 monocytes. So again, this is where your background, your prior knowledge, and your ability to spend time at your computer kind of come together to allow you to interpret your data. Last one that I'm going to go through is cluster 6, which we thought would be NK cells, and in fact, that's pretty unambiguous NK cells here. And then finally, cluster 8. So they said progenitors, but it's probably because they don't actually have platelets in the reference database that we were comparing to. Again, if we look back at our violin plots, we can see cluster 8 pretty strong platelet signal because PPBP is specific to platelets. So this is rough, and there's a lot of guessing and checking involved, but that's in many ways what the experience you have with single cell often is like. Ultimately, when you want to go to publish this, that's where you're going to really drill down and spend the time to absolutely justify every classification you made using literature sources and making sure you're doing it in the most robust way possible. But for a quick first pass through the data, this is totally acceptable. And hopefully by giving you these 3 strategies, you will be able to combine them and come to a pretty satisfying cell type annotation for your clusters. Hengi asked for the heat map. Is there any way to drop out the levels not used from the reference, since the total type of clusters are less than the types of labels from the reference? Okay, so I'm going to deal with that first before I get to your second question. So plot score heat map is just a wrapper around what appears to be p heat map. So you can just take this data here, this pred.mimd.fine. You can just take that and you can actually recreate this whole thing in p heat map. So as far as the plot score heat map, I'm not sure. But when you go to actually like publish, ultimately, you're not going to want to use the defaults from any particular package. It's really uncommon to see people publishing in high impact journals using like the defaults from something like scRNA or the default plotting from something like single R. So you'll probably need to go back and you know, do that the old-fashioned way in ggplot and pheatmap, and ultimately probably, you know, edit stuff in Illustrator to make the colors look really pretty and stuff. But it's up to you. It's up to you how you do it. As far as plot score heat map, you can examine the options and see if there's anything that will help you do what you want to do. But I wouldn't spend too much time messing with it, simply because it's probably best to focus on the actual interpretation for this first pass through. Okay, so second question: what if the cluster is more than one reference? Right. So are you asking about the situation like this cluster 0, where it had 480 CD4 T cells and 157 CD8 T cells? So it had 2 references in one cluster? Okay. Okay, so say you have 100 clusters. Okay, so if you have 100, so The reference will not cover. The reference will cover it. Yeah, so it's not annotating clusters, right? It's annotating individual cells. So it actually doesn't matter how many clusters you have. It's annotating individual cells. When you run single R, you're not actually telling it anything about the clusters. You don't give it any information about the clusters. The clusters are for your reference, but single R doesn't know about your clusters. You can actually make it know about them. That's another way to run single R, but I found it produces some weird errors. So it won't matter. When you go to plot it though, when you plot it, you can show the clusters alongside the label single R gave, and you can order it based on the clusters. So, if we have 8 clusters here, if you had 100 clusters, it would probably look kind of bad, but you would be able to see all of them here still in order. You can also order it based on the labels. So if I, okay, let me do this real quick. So if I take out these 2 lines, it'll order it by default based on the labels. Actually, I can just take out that 1 line. So now it's ordered it based on the labels rather than on the clusters. So we can see now our clusters don't look so clean, but the labels look very clean. Okay, so again, it's annotating the individual cells, not the clusters. It doesn't know about your clusters unless you tell it. You can actually do that, but again, I found it produces weird results, and I think it's more accurate to do the individual cells ultimately, because I think that's what it's optimized to do. Oh, okay. Okay. So you're asking if you have like lung cells in, okay, so if you have lung cells in your single cell experiment, but you only have a label set for neurons, then what are you going to do? Well, the nice thing about that is actually you do have a, well, okay, so 2 things. One, let's say that for the sake of argument, you actually do have a label set that covers neurons, because actually you do. Single R has, if you look at CellDex or single R, they have the same. If you look at the different databases available, you can actually see they have stuff from Human Primary Cell Atlas, which will cover all primary cells. Well, I shouldn't say all of them, but most primary cell types. We also have Blueprint ENCODE, which will cover pretty much all primary cell types. And so you can get pretty much any primary cell type you need for the most part. But let's say for the sake of argument that you only had neurons. What would happen if you tried to run this with your neuron reference, but you had lung in here? Well, it would still, so the way single R works, it's going to try to classify everything, and then it's going to spit out the highest confidence classification for each cell. And you're actually going to see in a second exactly what it looks like when you have that situation occur, when something isn't covered by the labels. So let me get to that in a second. Hopefully I'll answer your question there when we get to that. Because yeah, that's a great question. But I think you're about to see. Okay, so by going through by hand, looking at the predicted IDs and the square out clusters, and then guessing and checking, going back to my prior knowledge, and comparing these, I was able to determine that clusters 0 and 1 are probably, cluster 0 and 2 are probably CD4 T cells. Cluster 1 is probably CD14 monocytes. 3 is probably B cells. 4 CD8 T cells. 5 CD16 monocytes. 6 NK cells. 7 dendritic cells. 8 platelets. So it's a little bit labor intensive, but because it's such a central part of single cell, it's kind of important to learn it. Okay, so to actually add this back into your PBMC data set and to rename things based on the cell type you've identified for them, you use this rename idents function. But whenever you run that function, it's always good to save your new identities into a metadata column, or it's possible you might lose them if you rename your identities again. Because identities can change, but the metadata columns in general will not change unless you do something specifically to delete them. So that's why we run these 2 lines, and you'll see in a second why we needed to do that. Well, not in a second. After the activity, you'll see why we needed to do that. All right, so now when we run our dim plot, we can see here we have our labeled data set really nice here. And we can even take away the legend and instead use text covering each cell type. And if we wanted to, we could even compare this to, oops, sorry. We can even compare this to the clusters. So if you do group by scRNA clusters, and then you do plus dim plot PBMC, you can actually see them side by side if you want to. How can we show only one subset using dim plot? Are you talking about one subset of like the clusters, like one cluster? One cluster, yeah. Let's see here. So let's see. I know I haven't done this in a minute. So yeah, so there's a couple ways you can do it. So the first way, it's probably the easiest, is just to do split by clusters. But this might not produce the exact desired result you want. So let's just do this. Okay, so yeah, this can be a bit ugly. So it's now made, God, that looks terrible. It's now made a bunch of different UMaps, all sharing the same x-axis for some or same y-axis for some reason, with each of your individual clusters now separately. I'm guessing that's not really what you wanted. So let me show you another way, which is simply to choose the cells you want to look at. So there's a built-in function scRNA to do this, and I just don't remember what it is, to be frank with you. I don't remember. But actually, I think it's called cells. Cells by identities. They think everything. All right, so cells by identities is going to find the cells that have a particular identity. Okay, so let's say that we want to find the B cells, the cells in PBMC that are B cells. We run this, it's going to return the barcodes that are in the cells that are B cells. Okay, so it's these here. It's returning these cells right here. And we can actually supply that as an argument to dimplot. I'm pretty sure that allows us to make things look kind of nice. Oh, apparently not. Well, just like anything else in R, you can go to the help page. All right, cells. It wants a vector of cells to plot. Which, oh, right, because this is actually a list. So my bad. There we go. And now we're just seeing cluster 3, which we know is our B cells. What if we took our CD4 positive T cells? CD4 positive T cells. I think that was how we said it earlier. Yeah, that's not how we said it earlier. We called them CD4 space plus T cells. Well, that's not super helpful, is it? So now what we're seeing here is we're seeing the population we labeled as CD4 positive T cells grouped by their scRNA clusters, which are 0 and 2, if you recall. So hopefully that answers your question. Awesome. Yeah, that latent like plotting knowledge is coming back to me. I typically don't use scRNA's built-in plotting functions. I think it's nice to have your own because it's kind of limiting at some level what you can do in scRNA. But anyways, so for I know we're like out of time. So again, like if you got to bounce, do feel free to head out. But I want to give you guys 5 minutes to do what we just did, except using the mouse brain data set. So the reference that I want you to use is this mouse RNA-Seq from CellDex, which I guess you could also get from, let me just make sure that this works, single R, which you can. Okay, great. So you can also get it from single R. So go ahead, get this, do what we just did. And then what I want you to do, I'm going to give you 10 minutes actually. What I want you to do is attempt to prescribe the IDs to the cells, or sorry, to the clusters, and add them in and do your dimension plot. So it's a lot of work. I'm going to give you 10 minutes. Feel free to ask questions if you run into issues. But I think it's a really, really great real world example of the kind of thing you'll actually be doing. And this is a very real world data set too, by the way. We'll talk in a minute about what happens when you've got an ambiguous cluster that seems to have issues with it. So go ahead, we'll regroup at 7:13. And again, if you got to bounce, do feel free to do that. This will all be in the lecture later. Oh, and I just realized I didn't put the code from the last activity in there. Dang it. All right, 1 second. Let me also do that. So this was activity 1B. And there we go. This was the code from activity 1B, in case you missed it. All right. Okay. So I know that was probably very, very difficult, and 10 minutes may not even have been enough time to do it justice, but is anyone willing to volunteer? And oh, sorry, I was muted. Okay, so I know that was not a ton of time, and I know it's 10 minutes is probably not enough time to do justice to that assignment, but what I want now is if anyone's willing to volunteer to share, if you go ahead and put in the chat, if you're willing to volunteer to share your results and to share the cell types you chose. There's really no truly right answers here. This is partially subjective, partially based on your own judgment and biological background. But is there anyone here who's willing to volunteer to share that? All right, so I'm just going to go ahead and share how I would do this then. Again, there's really no like right answer, and this is probably one of the hardest things that in fact, this is definitely the hardest thing we've talked about so far. This is the least trivial part of single cell, the biological interpretation. So this is not easy at all. So if this felt difficult, it's because it is, okay? But that is why I tell you to copy and paste whenever you can, because single cell is hard. You shouldn't make more work for yourself without a good reason. All right, so I'm going to change SCEP to SCEB just to keep with the nomenclature. Simple find and replace. All right, so we get our mouse reference. We turn this into a single cell experiment object. We need to give it the mouse cell reference. So, sorry. Okay. All right. Sorry about that. Give it I clicked the wrong thing. Give it the mouse cell reference. Okay, so we've got our SCEB. And let's change MIMD to mouse cells. So one more find and replace. Again, just make your life easier when you can. There you go. Okay, and now this should be mostly good to go. Let's start going through it. So we run single R. Hopefully this didn't take too long to run on your computer. I know it's a lot of data now, so it can start to add up, especially when you get to the fine grain part. But okay, so it's already done. Hopefully it didn't take too terribly long on your computer. All right, let's go ahead and plot this now. Okay, so great. We can see here, just to reference back to what we've already talked about, if I go back to my brain violins, it's kind of a weird term. Cluster 0, as we recall, is probably oligodendrocytes. If we go to our heat map now, we can see it predicted cluster 0, probably oligodendrocytes. So we're seeing some good agreement between what we came up with from prior knowledge and what they discovered through this kind of unbiased sort of approach. And we can go through it, but I'm going to just do the fine-grained, and then I'm going to do the combination so that we can save some time here. So let's go ahead and do our fine-grained. Whoops. That is not what I meant to do. Dangers of find and replace. Sometimes you make a mistake. And all right, so we've now got the fine predictions. Let's go ahead and test those. Okay, so those don't look too terribly different. We can see some more granular data types like cell types like quiescent neural stem cells, stuff like that. So kind of interesting to see those pop up. All right, but now what we're really trying to do is combine these and get a more accurate global picture. Okay, so let's combine them and get that error message or warning message. I'm not really sure if it really means anything. I don't really think it does. And I'm gonna order it by cluster. So let's do this. And okay, now let's take a look at our combined results. All right, so we can see here in our broad scores, our fine scores, when you combine them, it looks like you're able to get some pretty satisfying agreement on which cluster, or so which cells have which cell type labels. So we're seeing a lot of stuff that's interesting. So we see oligodendrocytes. For some reason, it's put it twice here. Not really sure why. I'm guessing it's because maybe it's like there's a space at the end of one of them, so I didn't think they were the same. I don't know. I didn't make this. I didn't make the reference, so I'm not sure. Same thing with neurons. We can see here there's something strange going on in this cluster 4. It looks like it might be oligos, but it might also be NPCs or NSCs. We'll get to that in a minute. What that's telling us. And we can see pretty strong endo cluster here. Astrocytes. It looks like probably here, or perhaps NSCs. Fibroblasts here. Now we know there's no fibroblasts in the brain, or at least to my knowledge, there's no fibroblasts in the brain. That'd be kind of bad. But there are cells that are similar to fibroblasts, which reside in the brain. Specifically, they're called pericytes. And so we'll see in a second that when it's saying fibroblast, it's probably referring to pericytes because they're not too different from each other. So what do we do with this? Well, we can take our pruned labels and we can add that to this predicted ID column in our square out object. And now we can compare the predicted IDs from single R to the clusters. All right, so let's go ahead, let's take a look at this and see if we have anything that's kind of unambiguous. So cluster 0, pretty unambiguous oligodendrocytes. Like we said before, this one is, we already identified that as an oligodendrocyte cluster, pretty strong here. So cluster 0, oligodendrocytes. Okay, great. Let's move on. Cluster 1, pretty unambiguously for endothelial cells. If we look at our violins from before, we can see for endothelial cells that, in fact, they are pretty strongly in cluster 1, those endothelial markers. So we're pretty confident now to say that cluster 1, endothelial cells. Okay. All right, cluster 2. Now this one is less clear cut. We see astrocytes 45, QNSCs 134. Now this is where your background knowledge is honestly really important. So let's go back to our prior knowledge plots. Let's take a look at the astrocyte category here. We see our astrocyte markers are pretty strong in cluster 2, but there might be a little bit of ambiguity, at least in one of them. But GFAP is a pretty strong marker for astrocytes. It's a very commonly used astrocyte marker, which I know because of my background knowledge. Again, it's about your background knowledge to some degree. So I would probably say, even though we're seeing QNSCs here as well, I would probably argue that the reason we're seeing that is because QNSCs are actually transcriptomically kind of similar to astrocytes. And I would argue that this is probably kind of a false positive. It's probably more likely that the astrocytes are the true label here. But again, this is slightly subjective and slightly based on your background and experience. Okay, so in resolving this ambiguity, I'm going to side with astrocytes. I'm going to make that argument when I go to publish it, and I'm going to have a lot of sources to back it up when I actually go to do that. But I could still be wrong, and that's part of, that's the process with single cell. You can still be wrong. All right, cluster 3. This one's all over the place. And if I recall correctly, there were no clear markers that distinguish cluster 3 in anything we had here. So really not clear what that is, okay? We'll come back to that in a minute as to what's actually going on there. For right now, I'm going to put unknown here. Cluster 4, pretty unambiguously, it says ANSCs. However, I don't think that they're actually ANSCs. Again, prior knowledge is really important here. NSC, so that's active neural stem cells. These neural stem cells are not all that different from oligocyte progenitor cells, okay, or also known as OPCs. If I look at my oligodendrocyte progenitor cell markers, I can see, oh, wait, where'd they go? There they are. I can see they're pretty strong and pretty specific to cluster 4. So because I have this prior knowledge, I would probably side with oligodendrocyte progenitor cells for this one in my mind, but I'll keep the option open that it could be either, especially since this didn't seem to think it was, even though that label was there in the reference. So I would probably say OPCs or ANSCs. And at this point, it would probably take me a little while and a little bit of digging to really figure out which it really was. All right, 5. You can see here we've got neurons and NPCs. That's neuronal progenitor cells. That's a little bit of ambiguity, but I'd probably side with neurons since we're not in like a region of the brain that's going to be likely to have a lot of neural stem cells or NPCs in it. It's more likely we'll see the neurons here. So I'm going to side with neurons for number 5. And also, again, checking my prior knowledge, I can see my neuron markers pretty strong in clusters 5 and also 7. So 5, I'm going to say neurons. 6, it looks like it could be fibroblasts, which again, go back to your prior knowledge. Look at the pericyte markers. So pericyte, MCAM is a pretty strong marker for pericyte. PDG, FRB is another strong marker for pericytes. Both are pretty specific to pericytes. And we see those light up cluster 6. So I'm guessing that they're not actually fibroblasts because we really don't have any of those in the brain, as far as I know. They're probably pericytes. So I know I gave you 10 minutes to do this. If you didn't already have all of that background knowledge, this would have genuinely been impossible to do in 10 minutes. So part of this is to give you a chance to take your best shot, but realize that there are inherent challenges to this. And sometimes you have to take longer and get that background knowledge or find someone who can help you interpret it. All right, finally, cluster 7. So we don't need this cluster 8 here. There's not going to be a cluster 8. Cluster 7, we see neurons. That agrees with our prior knowledge, which showed pretty specific neuronal, highlighting the cluster 7 with the neuronal markers. So I would be pretty comfortable in saying cluster 7 is neurons. It's just interesting that it seems to be a different kind of neuron, or there's something else that's different about this cluster compared to cluster 5, which we also said was probably neurons. So now I've got my new IDs. That was the hard part. And I come over here. I can rename the scRNA IDs using my new IDs, save them in this cell type metadata column. And now I can plot. And now I can see them lit up within each of my clusters. I can also do it without a legend. And I can even do this with, okay, that's not what I wanted to do. I can even do this by adding them together. So with the original cluster labels. So I could do dimplot brain plus dimplot brain, and I can show, group by scRNA clusters to see how the clusters stack up to the labels. So clusters, labels. Is there a way to rename them? Okay, so renaming is, yeah. So remember when I said that you should save everything in a metadata column? This is what I was referring to. Okay, so let's say you want to go back to having the identities be the clusters, which is what you have here. When you rename your identities, the names in your vector, so this left side, these are the names of the elements of the vector, has to be your existing identities. So if I now want to rename this back, I would need to give it these, which are now the true existing identities. But that's only if I want to use the rename idents function. It's actually an easier way, which is I'm literally about to show. So let's skip all this because I need to do this in a second anyways. So the idents for PBMC right now are the actual cell types, B cells, CD4 T cells, etc. but I can reset them by finding that scRNA clusters column and just assigning that as the idents. And now it's the clusters are the identities. Let's say I wanted to go back and have it be the cell types again. How do I do that? Well, I would just do idents PBMC is PBMC cell type. This is why you needed to have that as a metadata column. If this was not a metadata column, if you forgot to create that column, you now have no way to get that again. You have to rerun the whole thing back from like the earlier step. So that's why you wanted to save this in a metadata column. Because as far as I know, there's no other way to recover that information if you want to then re-revert your identities back to clusters and then change them back to cell types. All right, so they're cell types right now. Let's make them clusters. So now my identities are clusters. Seems a bit arbitrary, but this is just how scRNA works. There's one more thing I was going to show you, which is if you didn't, there's actually a fourth way to get the markers, which is honestly not the best. So I'm not going to like belabor the point, but like, let's say that you wanted to, instead of, I'm just going to run this while I'm talking. Instead of using some prior knowledge that you got by hand, you wanted to use prior knowledge that you got automatically. Okay, so let's say you have a database of cell markers and you want to automatically annotate each of your clusters using that. Well, there's actually a way to do that. So the first step is to find all markers. So for every cluster, you will want to find the marker genes that identify that cluster. So these are the genes that are overexpressed in each cluster. Then I'm just going to ignore this for right now. So this is really just looking at the top markers in each. And if I make a violin plot of those top markers in each cluster, it'll probably look terrible. Oh, that's not too bad. I can see them here, but that's not really why we're doing this. So you can take a pre-built marker database like cell marker. So I'm going to show this here. I've got the URL is in the script. So if I just paste this, it takes me to the cell marker database and it takes me to the blood cells. So these are all these cells that are known to be associated with blood when we're dealing with PBMCs here. So these are blood cells we're looking for. And so what I can do is, okay, so I'll just back up. How did I get here? Well, cell marker, when you start, you're on this page. To get to those blood cells, all I had to do was search blood here. And it'll find all the markers associated with blood cell types. And there's a lot of them, and they come from experiments, they come from single cell sequencing, they come from literature reviews, and they come from company lists. Okay, so there's companies that actually publish these. All right, so in this particular case, what I did was I downloaded this list, okay, as a CSV, and that's actually what you have in your folder right now, cellmarker_pbmc.csv. So without belaboring the point, I can read this in, and I can do a bunch of fancy, data wrangling on it to make it kind of nice. Which you can honestly ignore this. I'm not going to go through how this is working. If you want to try deconstructing it yourself, you're more than welcome. If you want to ask me questions, you're more than welcome. But I'm not going to, for the sake of time, go through it now. Basically, what this creates is this PBMC_marker list data frame where you've got cell types and genes that are associated with cell types. We take this and we can combine it with our PBMC markers. So we just created these. This is, again, for every cluster, we know the genes that are overexpressed in that cluster. So by combining that with our marker list, we can see the cell types that are associated with the genes that are overexpressed in each cluster. And so this is a semi-automated way to get that information. Semi-automated because it won't tell you everything, but at least it cuts down on a lot of the work you have to do. All right, so final result is this. So I've taken the top 5 from each. So I only picked the top 5 markers for each cluster. So cluster 0, top marker, RPS27, associated cell types, multi-lymphoid progenitor cells, LDHB, naive CD8 T cells, and so on and so forth. Some of them are associated. So S100A8, which is a cluster 1 marker, is associated with CD1C plus B dendritic cells, lymphoid prime multipotent progenitors. There's actually a few that's associated with, but you can see them all here. All the ones that are associated with that particular marker are here. And so this is a way to kind of automate the process of gathering that data. I'll say this still requires a lot of effort because this is kind of like information overload. So it's hard to interpret and it just takes time. This is another thing you can combine with what you're already doing if you want. I wanted to just show you how to get that. Okay. So we're a little bit over time, as I'm sure you know. I'm going to quickly go through this because honestly, this part's going to go fast. Everything that we've already talked about is represented in this part. So we've identified what cell types belong to each cluster, but what if we wanted to ask more generally, what are the biological processes that are involved in each cluster of cells? Wait, I got a quick question here. Okay, so we can basically make any marker list by our own. Yeah, you can. You can absolutely do all of that. Yeah. So if you have your own list ahead of time, if you have your own prior knowledge, you can use the same code with it. It's just the wrangling might be different because some of this wrangling is specific to the format that they output in the CSV file from the website. So you might have to change the wrangling a little bit, but for the most part, it'll be identical. Okay. But again, this is probably not like the best, honestly, not the best approach. I think this is probably out of all the approaches I tried all 4 approaches while I was making this lecture. This was the one that I found to be the hardest to actually use in practice. All right, so when you do your cluster marker analysis, you're trying to understand not what cell type each cluster is, because you kind of know that now, but what are the biological processes that those clusters are participating in? The cells in those clusters, what are they doing biologically, besides just what kind of cells are they? So again, this is probably more relevant most of the time when you're dealing with something like tumor data and you've got a bunch of clusters in your tumor and you want to figure out what is each different tumor cluster doing. But for right now, we can actually see this exemplified by the data sets we already have to some degree. So to actually do this, we're going to run that find all markers command, which I've actually already run here. I think. No, I didn't. So run the find all markers command. So it's going to find the genes that are overexpressed in every cluster. And then we're going to filter for the significant markers that have a p-adjusted value of less than 0.05. And we're going to write that to a CSV file because we're just about to show you how you can use Enricher, which is an online tool, in order to analyze these. So, okay, what did I do? I wrote that to a CSV, opened it up in Excel. This is literally how I do this. Like I know R pretty well, and this is still, oh, that's not what I meant to do. This one, PBMC sig markers, my bad. I know R pretty well, and I still prefer doing things like this when I'm doing the first pass through the data, okay? All right, so let's say we want to figure out what are the biological processes that are upregulated in cluster 0, okay? So we believe that this is a CD4 cell cluster. So let's go ahead, filter. Hopefully you all know how to do this in Excel. You can filter this column to only show cluster 0. Okay, so now I'm only showing the 84 genes in cluster 0. So I can take these, Control-Shift down, lets me select all of them, or you could just drag to select them all. Go in your browser, just Google Enricher. First thing that shows up, Enricher from the Mayan Laboratory, really fantastic group. Paste your list. If I'm putting this to chat now. So I just put the link to Enricher in the chat. So if you didn't see that, you have it now. Okay, so paste your gene list. I got my 83 genes here. These are the 83 genes that were overexpressed in cluster 0. Let's now see, what are these genes doing? All right, and it was fast. So we've now enriched for over 150 databases that contain pathway level and gene set level information. So let's start with what we already kind of know. So Enricher, just for the uninitiated, Enricher is kind of information overload. It's super powerful, but it can be really tough to sift through and find something worthwhile. So let's start with something that we already know we should see. Let's look at our cell types. We know that these should be CD4 cells, CD4 T cells. And we see right here, human gene atlas, CD4 T cells, P adjusted value of less than 7 times 10 to the negative 14th. So quite significant. So this makes us feel pretty confident that in fact, these are markers of CD4 T cells. So now that we know that, and we also see that here, and we see other, immunological things here, let's now take a look at the different things that are contained in, okay, Mauricio, I forgot the code for 1C. I'm so sorry about that. Thank you for reminding me. I'm going to forget that every single time. I'm certain of it. Hopefully this will all paste. I know there's like a limit. There's a limit. Okay, so that was part 1. I'm going to put part 2 starts at this line 295. Sorry. There's like a weird text limit. So anyways. Yeah, so yeah, that should be fine then. So when we're interpreting Enricher, I'll tell you how I do it because I use Enricher many times a week, every week. And I find it to be an incredibly powerful tool. So I start with the transcription factors. Now, these transcription factor databases, what they did was they found where do certain transcription factors bind in the genome? What genes do they bind to? Those genes become the gene set for that transcription factor. Therefore, MYC-CHEA is the genes that MYC is known to bind to from the CHEA database. Okay, so that's what we're seeing here. So the fact that I'm enriching for these genes in cluster 0 might tell me that maybe I'm seeing an overexpression of MYC targets in my CD4 positive T cell subpopulation in cluster 0. Kind of interesting. I don't know if you guys are immunologists, but I thought this was super weird when I first saw it. And then I Googled, MYC, and what did I do? I did CD4 T cells MYC. I Googled that. And actually the very first thing that came up was an eLife article showing how MYC controls T cell proteasome. So actually this connection might be pretty well understood at this point. It's just really cool because I wasn't expecting to see that. So pretty interesting stuff. Again, do exercise caution when you're interpreting Enricher because it's very easy to see what you want to see, right? This is something that a lot of people don't say in like workshops and stuff like that because they're not telling you what it's actually like to work with this kind of data. But I'll tell you, like, it's very easy to look at this and see what you want to see. So always be skeptical of your own results. Another thing that happens is the more genes you put in, the more likely it is you'll get significant results. So Enricher is a tool just like any other, but it's got its limitations. So do bear that in mind. Anytime we do bioinformatics, we should be doing external validations as much as humanly possible. But then again, when I see MYC here and here and here and here and here, I feel more confident about it. So it's partly, your mentality of approaching the data and partly, the actual objective data itself. All right, so now let's move on to the pathways. Pathways now these are gene sets that describe some biological process that has a certain number of genes that are associated with it. So for example, KEGG is a really commonly used pathway data set. If I take a look at the KEGG results here, I can see the top result by far is ribosome. Its P adjusted value is less than 1 times 10 to the negative 66th. Very significant. And then following that, I can see hematopoietic cell lineage, which makes sense, Th1, Th2, which also makes sense because these are CD4 T cells, and lots of other stuff here that I expect to see. But ribosome, I wasn't actually expecting to see. I go back to my data now, I take a look at it, and I say, all right, why is it giving me that result? If I look at, well, I'll do it in the Excel. If I look at the actual gene list I've just put in, I can see there's a bunch of RPS, RPL, RPL. Those are actually ribosomal proteins. So it makes sense actually that because there's so many RPS genes and so many RPL genes, that the top thing I enriched in this list was actually ribosome. So this is, how do you interpret your data? How do you make sense of it? So lots of ribosome translation, which relates back to ribosome, a bunch of stuff relating to ribosome, relating to MYC targets. So this all kind of makes sense. I'm not going to dig through it any further at this point. Let's take a look at our ontologies. Now, ontologies, Go is an example of an ontology, are structured. So it's not just a pathway. You actually have how do pathways relate to each other, which there's benefits to that. I don't usually use the hierarchy when I'm looking at it like this, though. So we actually see CD4 T cells. So again, more validation that this is what we're enriching for and that this is correct. See more ribosome, protein, ribosome stuff. So yeah, pretty strong ribosomal signal. Diseases and drugs, I don't typically spend too much time here because they're honestly kind of hard to interpret. And I wouldn't really expect to see anything here anyways. Cell types we already talked about. Miss, I don't typically spend any time here. Legacy, don't spend time here. Crowd is probably the most interesting and the hardest to interpret of all of them because this is like a wild card. You can find a study from 10 years ago that happened to find the exact same gene list you just found in a totally different biology. And you can go back and read that study and you can see, oh my gosh, they're actually seeing the same thing I'm seeing, but they're seeing it in breast cancer. You know, it's really cool to see that. And this is how you can connect what you're seeing to the larger world of biology. But it can also be information overload, hard to interpret. So it's worthwhile to just spend time here. All right, so I promise I'm not going to spend much more time on Enricher. I just want to show you how to actually do this from R. Okay, so first thing, once you do this in the web browser, you can actually get a permalink to this. So from now on, I can always revisit this analysis by following this link. They'll keep this link active. And so I can just always come back to this analysis even after I close my browser. Super cool. Love that feature. Mayan group is amazing. Let's show you how to do this in R though, because let's say, you want to make like a really nice GG plot or something. So let's go now here to Enricher. So there's actually a package called Enricher. So this is just the R interface to the Enricher web application. So you can do all the things you can do the web application in R. So the first thing we can do is get the databases that are in Enricher that are available to us. Let's close a few of these because it's starting to get crowded. And that's this DBS object here. And so these are all the different databases that are available. So we can see we already saw Human Gene Atlas, we already saw KEGG, BioCarta CHEA, and there's literally over 170 of them now. So many. It's really maybe too many. But let's say we want to just focus on 4. We want to get the Human Gene Atlas. Let's say we want to get our ENCODE and CHEA transcription factors, our KEGG, and our MSIG-DB hallmark gene set. So if anyone knows GSEA, the same people made the MSIG-DB, and they made this hallmark gene set, which is really useful for just seeing broad biological processes. So let's get our cluster 0 genes. So we take our significant markers, filter to get cluster 0, and then we pull the vector of genes out. And these are our 83 genes that we've now got in here. And now all we do is put them through the enricher function with our databases that we said we wanted to check. So hit, whoops, I forgot to run that line. And it's done. It's very, very fast, especially when you've got a small number of genes, it's very fast. I built a custom function to do the plotting. So you can deconstruct that if you want and change it and make it look nicer because it doesn't look the best, but at the very least, you have it here at your disposal. So I'm going to apply the plotting function to each of my databases. So by doing the enricher, we got this cluster 0 eres list object. So so this is a list containing for each database a data frame with the results. So if I just view this first one from the Human Gene Atlas, I can see it's got the CD4 T cell term, has the lowest p-adjusted value. This is the combined score from it. So that's just another metric of how significant it was. And these are the genes from my list that I uploaded that were also found in that CD4 gene set. So that's what the genes are. They're the genes you uploaded to Enricher that they found in the gene set. So these genes are in my list and they're in the gene set called CD4 positive T cells. So I want to plot this. And I want to plot each of these individually, and I want to combine them into a nice plot that I can examine and have fun showing my boss. Okay, so how do I do that? Well, I made my function essentially to do that. I apply that function across each of my results here, and I collect them, and then I can arrange them using cowplot into a matrix of plots. And you'll see in a second what this produces. Okay, so I just produced this and I actually saved it to this file cluster0_enricher_plots.png. And that shows up right here. So let's take a look. And there you have it. So we've got our 4 databases. We checked Human Gene Atlas, ENCODE, KEGG, and MSigDB. We've got the top results from each ordered by p-adjusted value and then colored by the combined score, which is just another metric of how significant it was. And just like we saw before, Human Gene Atlas, CD4 T cells, KEGG, ribosome, MSigDB, MYC targets, and interestingly, I didn't even see this before, allograft rejection. That's kind of curious. And MYC, CHEA, and MYC, more stuff. MAX, which is just another way of saying MYC. Okay. So that might be something I would show like to my boss or something like that. Okay. All right, we're almost done. Thank you for those of you who have stayed on this long. I don't know how you did it, but truly, if you need to leave, you can leave. We're really just going to be rehashing what we just did in different ways. All right, so as we recall, we've got our clusters and our cell types. So what we're going to do now is we're going to find for clusters 0 and okay, so let's ask a question, right? So CD4 T cells was the annotation for both cluster 0 and cluster 2, but there's got to be something that makes them different from each other, right? Let's see if we can figure out what that is. So what we're going to do is we're going to pull the significant marker genes from both cluster 0 and cluster 2 into a cluster gene list. And so now we've got cluster 0 and cluster 2. We've got our markers. So 83 for cluster 0, 49 for cluster 2. And we can name it so it looks a little bit nicer. Cluster 0, cluster 2. Now what we can do is we can use the Venn diagram package to just directly compare them. What were the genes that were shared and what were the genes that were unique to each cluster? And that's it. There you go. So that should have created a PNG called clusters 0-2 compared. And there you have it. So there were 17 that were shared between them, but there were a whopping 66 that were unique to cluster 0 and 32 that were unique to cluster 2. So there are some pretty big differences. And if we want to see what was in each of those overlaps, we can just use the calculate overlap function. Take a look at that overlap list. And we can see now the 17 that were shared. CD3 kind of makes sense. A few other things that kind of make sense here. But what was unique? Well, for a start, we can see here in the ones that were unique for, just to show the Venn diagram again, the ones that were unique, the 66 that were unique to cluster 0, we can see that includes a lot of our ribosomal proteins. Very interesting. We can also see here that the ones that were unique for cluster 2 include some things like IL-32 that might be more of a maybe more activated T cell marker, kind of hard to say, and maybe some things that have to do with interferon. So, but again, we don't have to look at this by hand. We can upload this to Enricher and we can check. But there's actually even a better way than all of that, which is to, in our original calculation of our marker genes, instead of just finding the markers for each cluster, let's now directly pit cluster 0 versus cluster 2. And we're going to use our differential expression model to robustly find the genes that are specifically differentially expressed between these two clusters. This is more accurate than simply doing an overlap with like Venn diagrams. This is going to give you much richer information. And to do that, you just set ident1 to be the numerator, and ident2 is the denominator. You can reverse them. It doesn't make a difference. The only thing that's going to impact is the sign of the fold change. So if you reverse them, it would just reverse the sign of the fold change. The p-values are not going to change at all. So go ahead, run that, and it's done. So let's go ahead and take a look at that now. And I call this cluster 0 versus 2 markers. So that's right here. So let's sort it by our log fold change first. So these are the top that are overexpressed in cluster 0 compared to cluster 2. CCR7, mallet 1, some interesting stuff there. Let's now look at the top underexpressed by sorting the other direction. So now we can see S100A4, L-Gal S1, some interesting stuff here. All right. Oh, I froze. Okay. Now let's actually pull those genes. So I want to know what are the genes that are overexpressed in cluster 0 compared to cluster 2. So these are the genes that are very specific to cluster 0, and they're not going to be found in cluster 2, or they're going to be underexpressed in cluster 2. And so here's those genes. A lot of RPS, RPL, kind of interesting, some other stuff here. But let's actually throw that into Enricher, and let's see what our results look like. Making our plots. Let's now take a look at the plot we found. Cluster 0 versus 2 Enricher plots was the object we created. Okay, great. So now we can see here, top hit from human gene atlas was actually CD4 T cells, but if you look at the negative log 10 P adjusted value, is not significant. Okay, so nothing in here was actually significant, which is good because that would be kind of bad if we thought they were both CD4 T cells and one of them wasn't. They're most likely just different kinds of CD4 T cells. Other stuff we saw MYC, a lot of MYC. So perhaps this is a particular type of CD4 in cluster 0 that has a lot of MYC activity. And ribosome was very strong p-value. So high ribosome, high MYC activity. If I was an immunologist, I'd be able to tell you exactly what that is. I'll say what Swearot thought it was, because Swearot actually took a shot at identifying these. So if I look at that Swearot tutorial, where it actually tries to find the cell types, it thought that they were in cluster 0. It thought that they were naive CD4 T cells. Okay, so something about that gene expression profile to them said naive CD4 T cell. But again, I'm not an immunologist, so really I can't say confidently. It would take me a lot longer to say for sure. So now let's look the other way. Let's see what is overexpressed in cluster 2 compared to cluster 0, which is really just we take what we just did and we reverse the log fold change the other direction. So we have a lot more genes this time. So 242 are specific to cluster 2 as opposed to 40, which are specific to cluster 0. We do our enrichment and we're done. And let's go ahead and do our plots. Okay, so now we're seeing some interesting stuff. We're seeing some interesting kind of cell type profiles. Not really sure even what to make of these. Whole blood, no idea how that got there. Again, there's limitations to doing this kind of analysis. So this might be some, fluke of how we're doing it, or it might be that the whole blood gene set actually overlaps with something that we're currently finding. It's just the labeling is not very informative. We also see interesting things like CD33 myeloid and CD14 myeloid and CD56 NK cell. Really just not sure what that's referring to. Hard to say. Let's look at some other things here. So I'm going to just jump down here to the bottom. So we're seeing some things that kind of indicate a stronger infection phenotype. So perhaps something that is more aggressive, a T cell that's more aggressive. So we see shigellosis, actin, antigen processing presentation, viral myocarditis, phagosome. So things that are potentially indicative of a more active infection phenotype. So potentially these are more of an activated CD4 T cell as opposed to a more dormant one or naive one. And that's actually, again, in line with what Swearot said when they went through this data. If we look at the MSIG-DB, this is echoed by the fact that some of the top hits are actually interferon responses. So potentially indicating that you have a more intensive immune activation occurring in this population. So again, back to what Swearot said, it said that this cluster 2, that they are memory CD4 T cells as opposed to naive CD4 T cells. So we actually do expect these to be a bit more activated and to have some of those kind of differences that we saw, at least to some degree. If I was an immunologist, I'd be able to explain it better. I'm not, but hopefully that illustrates how one would go through this. And those who had that background would probably be able to do an even better job than I'm able to do here in explaining it. All right, so I was going to have you do this activity. It is now way over time, so I'm not going to have you do it. I would do this for homework. Basically, we're just repeating what we just did, all that pathway enrichment, all the cluster enrichment, doing that on the clusters within our brain data set. Okay, and so I'm going to do this before next time. And at the beginning of the next lecture, I will show my answers for this. So this will be something to think on over the next week. And hopefully this feels, if not easy, because it probably won't be easy, at least it feels doable. Okay, with that, thank you all for hanging on so long. I'm just going to ask, do you guys have any quick questions before we break? Okay, well, thank you all so much for coming and for staying so long. I know we went way over time, which we're probably going to keep doing, to be honest with you. There's just way too much material, and 6 weeks was a bit ambitious on my part. So I apologize about that, but hopefully, if you do have to leave early, hopefully at least the lecture recordings are giving enough of that information to make sure that you're able to get the material. So thank you all so much for being here, and yeah. I know this was probably the hardest lecture so far, and this is the most difficult part of single cell, in my opinion. But, you know, if you can learn to do it well, and if you can learn to feel comfortable with the process, there is really no limit to what you can do with single cell. So, all right. Thank you all so much, and I will see you all next time. Have a great week.